Ladies and gentlemen, welcome to this Aelis Farma web conference regarding the phase 2B clinical results in cannabis use disorder. I'm happy to have with us Mr. Pier Vincenzo Piazza, CEO of the company.
Hello.
And Ms. Stephanie Monlezin, Chief Operating Officer of the company.
Hi.
Before handing over to the team, I'd like to inform you that you can ask your questions in written form through the chat system at the bottom of your screen. And there will be a Q&A session at the end of the presentation. I now give the floor to Mr. Pier Vincenzo Piazza.
Okay, thank you very much. And first of all, welcome everyone, and thank you for taking the time to discover these results with us. I would like to say as an introduction that we are communicating the results because given that we are a public company, it's an obligation to give you guys an head up as soon as possible. But we have not completed all the analysis and the QC, and so these results, for some of them, can be considered preliminary in some sense, even though I don't think there will be many surprises once we have completed the process. Okay, so usual disclaimer, and I would like to start going over again on the design of the phase 2B. So this is probably the first large-scale multicentric pharma-sponsored study in CUD, exploring primary binary endpoint. I will come back on that, what they are. Professor Frances Levine was the coordinating PI at Columbia. It's a double-blind, placebo-controlled study that has included 333 patients with CUD in 11 clinical centers, both academic and professional centers. We have tested 3 doses of IF versus placebo, and the treatment has been administered once a day for 3 months. The major objective, independently from the primary endpoint, was to identify what were the primary endpoint that could be best suited for next clinical study, and also what was the most effective dose of IF 117. So clearly we have looked at the safety and tolerability endpoint, typical treatment, emergent adverse event, but also vital sign and safety laboratory parameters. We had also some pharmacokinetic endpoint, see-through, that is basically the baseline of the drug, and the evaluation of the time to steady state and what's happened once we reach the steady state, and the clearance of the drug after treatment discontinuation. So, as I said before, the main endpoint, including the primary endpoint, were binary measure of cannabis use. So a binary measure is a measure that evaluates the proportion of participants that meet a predetermined criterion of days of use per week within a predefined time window. And once they meet the criteria, they need to stay to that criteria all the time. So we had 1 day of cannabis use per week, 2 days, and 0 days that is complete abstinence. We'd also had several quantitative endpoints measuring cannabis use and cannabis use related behaviors. So, for example, the number of days of cannabis use per week, or overall the treatment period, the amount spent on cannabis use per day of use, and the concentration of the THC metabolite, THCCOH, that is basically the only objective measure of the study, because all the rest are patients reported or clinician-made evaluation. So we also measured quality of life, quality of sleep, and anxiety and depression using the Hamilton scale. So let's look at the results. Safety, I would say that 117 confirm the good safety that is observed with this compound, but it's a characteristic of the class of drug, the CB1-SSI, to which this drug belongs. So the drug was very well tolerated and no safety concerns were observed. And the type and frequency of adverse events were similar across all treatment groups, including placebo. So this is good news because this is the longer study we have done with a drug of this class. It is important to confirm this quite good safety and tolerability profile. The PK, nothing unexpected, so we found a very good dose linearity. The C2 concentration were in the range of the predicted value, even though they were a little bit higher, but still in the range of what we had predicted. As a reminder, we have a POP PK model that allow us to make some prediction on the behavior of the drug. An important point was to confirm that the predicted steady state by the model would be reached in the same range and was 28 days, exactly what predicted. And once the drug reached the steady state, we don't see any accumulation. On the contrary, once we discontinue the treatment, as expected, and with the expected speed, the drug concentration drop. Then we have the results with the binary endpoints. They are the ones that are disappointing because we didn't find any statistical significant effect, but what we found was kind of surprising is when you use this type of measure, the number of participants that reach any of these binary criterion, even the placebo arm, are very low, which indicate that this type of measure is probably not adapted because it's insensitive to measure cannabis use in this clinical population. So the conclusion of this is the binary endpoint do not seem what we should use when doing a trial in CUD. Again, this was kind of unexpected, but this is the first really large trial, well controlled, that allowed us to make this type of discovery. On the contrary, when we look at quantitative endpoint measuring cannabis use and cannabis use related behavior, we find with the highest dose of IF 117 is 1 milligram, almost statistically significant improvement in quality of life, anxiety and depression, and almost statistically significant, this quality of sleep, in quality of life, we had a trend. This finding is important for the pharmacological class because, as you know, the CB1-SSI are inhibitors, and the major side effect of previous CB1 inhibitors, they are antagonists of the previous generation, like rimonabant. Their major side effect is an increase in anxiety and an increase in depression. And so seeing that in a long trial, instead of increasing this parameter, we decrease them for anxiety, the change is significant, is a very good news for this class. That means that if we want to get rid of the effect of CB1 antagonists, not so much brain periphery type of thing that we should do, but probably the best approach is excise molecularly the side effect as our class of drug allowed to do. We also found consistent trend to a decrease in cannabis use, which become statistically significant in participants with moderate COD. And that is a very important fact. Why? Because this showed that the results of the phase 2b are consistent with the results of the phase 2a, in which only quantitative endpoints or variables were measured, and most of the patients were moderate. To illustrate this statement, I have a slide that shows you the comparison between the two studies. So the population of patients in the phase 2a, they reflect perfectly the epidemiology of COD, because we had 80% of moderate and mild, with a majority of moderate, and only 20% severe. In the phase 2b, for reasons that right now we are still investigating, we end up with 82% of severe. And it's not something that we could have controlled, because the inclusion criteria were almost identical in the two studies. So maybe one difference is in the recruitment method, because many of the patients of the phase 2a were recruited on a group of patients with which Columbia was used to work, and the phase 2b was done by advertisement in the newspaper, in the metro, and then the patients would call the center. Another difference is an impatience towards outpatient study. Clearly, in an outpatient study, we can see probably more moderate effect, because the patient is not in a controlled situation. But the important point is that in the phase 2a, we didn't study any binary endpoint, while binary endpoint were the primary endpoint and the main focus of the phase 2b. So why we didn't did it in the phase 2a is because you need a huge number of patients, like the one we have in the phase 2b, that are close to the patients you normally see in a phase 3, to be able to measure a binary endpoint. We had in the phase 2a, like in the phase 2b, quantitative endpoint, subjective effect of cannabis. Clearly, subjective effect cannot be studied in real life conditions, because you need to put people in the lab. But we also measure cannabis use by evaluating cannabis self-administration in the lab. And what is very encouraging for us is that we find a statistically significant decrease in the amount of cannabis used over the trial and per week in participants with moderate COD. So actually, the two studies are predictive one of the other. So what are the main takeaways? The first, and I would have liked to do this, to know this before starting the trial, but again, is one of the problem of being an innovator, is that you have to break all the ground. So binary endpoint do not seem adapted to evaluate change in cannabis use in COD, because with binary endpoint, we had practically no measurable effect even in the placebo arm, that demonstrate that the measure is insensitive. So for this endpoint, this trial should be considered more a failed than a negative study. For the ones that are not familiar with this term, what is the difference? A failed study is a study that is not able to measure what was aimed to measure, and a negative study is a study that has been able to measure what it was meant for, but has found no effect. An important thing that the trial has revealed is that quantitative endpoints seems the best approach to measure cannabis use in CUD, because it's where we see consistent trend, some statistical significant effect, and these are the data that we are more controlling and running new analysis now that we know where we have to look. Another important element for the company and for the pharmacological class that we are developing is that this study further validate our new pharmacological class, the CB1-SSI, because the fact that to find IF117 pharmacologically active in the phase 2B, having an effect that go in the good sense, and it is in agreement with the effect that we have seen in the phase 2A, is really an important proof to demonstrate that these drugs that they have a mechanism of action that have never been tested in humans before actually works. Now what we are doing, we are investigating the quantitative improvement observed in phase 2B to further determine the best strategic and regulatory course of action for IF117. I would just give you also an update on the other thing we are doing. So the first good news is that we have cash up to the end of 2026 that give us time to develop a lot of things and also see for a partner to continue with IF117. This partner could be Indivior. They will make a decision after seeing the analysis of the complementary data. We will have the results of the phase 1-2 in people with Down syndrome with our second compound, IF217, in Q4 2024, and we plan to launch a phase 2 during the first semester of 2025. For the people that follow us, we also have several new compounds with innovative effect and broad perspective. And today we have 2 development targets: peripheral disease, and in particular obesity and related metabolic disease, because now we know that even with long treatment, we don't have the side effect of typical CB1 antagonists. Another interesting target is attention deficit hyperactivity deficit disorder, ADHD, and in a more large way, the condition that depend on a hyperactivity of the dopamine system. And I thank you for your attention, and again, thank you for attending, and we are ready to answer your questions.
Thank you, Pierre. I remind our participants that they can ask questions in written through the chat system. We have some first questions. First one, how confident are you about the future of CB1-SSI compounds given these phase 2B outcomes in cannabis use disorder?
So as I have said during my presentation, and I'm sorry if that was not very clear, this study is disappointing for its primary endpoint, but it's good news for the entire class of compounds. And so we are very confident. Another thing that you people should realize, when you study addiction, you are basically measuring endpoint that involve the willingness of the person to change behavior, or the ability of the person to change behavior, while in all other conditions where we are moving with the other compound, what we measure are functional improvement. They are quite easier to reach. So having the class validated and knowing that we have easier measure to reach is really good for us.
Thank you. Perhaps another question regarding the dose tested. The 1 milligram dose was the highest one. How was it determined, and would it be perhaps useful to have tested higher dose?
So it was determined based on the dose that give the highest effect on the phase 2A. It was also determined based on the fact that the 1 milligram after 3 months of treatment would reach higher concentration than what observed in the phase 2A. And so to the question, should we need probably to use higher dose? The answer is probably yes. But what is good is that we have such a good safety profile, safety margin, that is not a problem.
Thank you. We have no questions or additional questions so far. So I remind you that you can ask questions through the chat system. Another question: how long do you think it will take to have more deep data analysis?
Before the end of the year. I hope that it's going to be much faster, but I don't want to promise things that we are not 100% sure to be able to deliver. So we know that we can deliver before the end of the year, and the moment when we have clear results and we are sure about the database and all this very complicated statistical stuff. Just to give you an example, is we have two, normally in a study like this, you have an independent statistician. We have two in order to be sure of the results. So it will take some time, but before the end of the year, that's sure.
Thank you. And sorry, I have another question just right now. Could you repeat, please, the insights you learned about this study and its results regarding subset analysis or subgroup?
We didn't get you. There was a problem with the network. Can you repeat the question?
Yes, of course. So we have a question, which is, could you repeat the insights you learned about this study and its results regarding subset analysis?
The insight we learn about this study and its results. Okay. So the insight that we learn about the study are twofold. First, that binary variables do not seem very sensitive. I would say it seems impossible to use in a CUD population. We have also learned that quantitative variable can be used because we see a general trend and we see a significant trend, but not a significant effect in people with moderate CUD. I would like to remind everyone so that with the DSM-5, drug use disorder are classified in 3 categories of severity. All these people have a drug use disorder, but it can be mild, moderate, or severe. And in cannabis use disorder, normally you have 20% of severe. That is pretty different from other addiction where the number of severe is much higher, like, for example, opioid addiction. So we have learned that maybe with cannabis you should intervene earlier before the pathology become too severe. I would like to stress that in medicine, this is the rule. I mean, no one waits for a cancer to be metastatic everywhere before starting treatment.
Okay.
And there is, in general, in medicine, we try to start treating, this is true for diabetes also, we start treating patients when the disease is not severe. The advantage with CUD compared to other addiction, the not severe patients are the majority of the population. And this population, all the CUD population, is pretty huge because last data in the US show that in a population 12 years and older, 7% of these people met criteria for CUD. And that is 19 million of persons that today in the States met criteria of CUD. So I think that this is becoming so big that the idea of starting treating people before the disease become too severe, I think is a sensible idea, and that we leave the compound with a very consequent market.
Thank you. Meanwhile, we had another question. There was no placebo effect on binary endpoints.
Yeah.
In what way is this an indication that binary endpoints are not suitable?
Well, in general, when you want to use a measure, you need to have a signal. And in psychiatry, in general, you always have a signal. Even a placebo effect is a problem because you always have a very strong placebo effect. So seeing a measure that do not even detect a placebo effect, I think show that the measure is insensitive to this type of population, and they cannot really be used except if now you do trial probably with a huge number of subjects, but then it's not feasible.
Thank you, Pierre-Louis. New question. Are you considering other binary endpoints in this phase 2b study? And if so, what are they and how do they align with the primary study objectives?
We are performing, as I was saying, supplementary analysis. So we are looking at less strict endpoint, but I don't have those data yet. So we need first to do accuracy and be sure what is in the database. These are complicated studies to have very reliable results. So again, I'm sorry to not be able to show you quantitative data in general on all endpoint trends that we may have, but again, we wouldn't be a public company, and we would have a policy in our company of complete transparency with the public, with our investor. We would have delayed this webinar at least of 1 or 2 months in order to be ready to communicate once we knew exactly what we had. I think that is important, even though it could seem confusing, and it's our duty to give you what we have right now. And I hope that you will be patient enough to wait for the complete set of results.
Thank you, Pierre. This was last question so far.
Okay.
No other questions coming. So,
Okay.
Perhaps one last just came in here. What would be a potential time frame for the company to generate a drug candidate for obesity? Are you having BD discussions on that?
Yes, we have, as we have discovered, as we have described, these are discovery project, and so you will take. So I mean, it's not an easy question to ask. If you mean to say how long it will take us to show with preclinical data that we have a potential candidate for obesity and metabolic disorders, that could be pretty quick, even though with preclinical data, you cannot actually predict exactly. And then to go in humans, that will take few years, because just all the non-clinical work that you have to do to go from animals to humans take 2 years at least. Okay.
Thank you. Very clear. No more questions so far.
Okay. Then we thank everyone for attending, and please stay tuned for future news. Thank you.